Ooh, I'm not sure what I did. All right. Hello. All right, we're gonna talk about cell cycle. I'm gonna start with chapter two, right? Cell cycle, pretty easy. G1 S phase, G2 M phase. G1 S and G2 they're considered interphase, and then you have M phase, right? In G1 restriction points, you create organelles and protein for energy, and you increase the size of the cell itself. The S phase, you replicate the chromosome into sister chromatids, right? So you would have twice as much DNA as you had in G1, meaning that you would end up having 92 chromatids, but the ploidy didn't change, right? The ploidy didn't change. The ploidy of the cell didn't change. G2, it's a quality check point. That's what the book says. M phase mitosis. We're gonna talk about mitosis later. Uh, control of the cell cycle is between G1 and S, G2 and M, and then the book talked about the importance of P53, right? Just to remind you, P53 is a tumor suppressor gene. The gene is TP53 is the gene of P53. P53 is a protein. The protein P53 has to be tetramized to become abundant, right? But there has to be a stressor. Stressor stabilize P53, then become abundant, right? Then P53 can cause apoptosis. It can stop the cell from repair, right? Repair apoptosis, right? Um, Cyclin and CDK, they're also important for the cell cycle. Specific cyclins have to bind to specific CDKs for the cycle to move forward. You do not have to know, I don't think you have to know the specific cyclins of CDKs because the book didn't mention it, but we learned them in cancer. So if you want to learn them, just go back to your notes. Cancer, right? Cell going out of hand. Proteases are a big part of cancer, right? Proteases, if you remember in cancer, uh, MMP could trigger proteases, right? Production of proteases. Proteases are crucial for invasion of basement membrane and promotion of angiogenesis, right? Those two are two of the main factors for metastasis of cancer, if you remember. All right, mitosis. I actually drew a picture. Interesting, right? I drew a picture. Unbelievable. I drew a picture. Uh, Give me a sec. What is my picture? Well, hi, right there you go. All right, first we're going to talk about mitosis, right? I'm so sorry. Mitosis results in two identical cells created by a single cell. So this is a single cell created by a single cell, ended up with two identical cells. Identical, keyword identical, somatic cells that are not involved in sexual reproduction. All right, that's about prophase, right? And this is prophase. So this is how it looks like an interphase, right? G1, S phase, and G2. The chromatin is decondensed. You have a nuclear membrane around the chromatin. You have centrioles. So in prophase, what happens is you have condensation of the chromatin, right? So these decondense become condensed, right, forming a chromosome, right? Chromosome is the condensed form of the chromatin. Chromosome is the condensed form of the chromatin. Okay, remember that. Centrioles pair separate and move towards opposite poles, and the centrioles have spindles. So, as you know, as you see in interphase, this is the centrioles. They didn't have the spindles made out of microtubules, but in prophase, you have centrioles and you have microtubules, right? And what is more? All right, that's it. Let's move on. Metaphase. Metaphase, this phase, right? So you have, so let's say this is the chromosome from your dad, this is the chromosome from your mom, and centrioles, right? Each centriole is pulling one of the sister chromatids from your mom and your dad at the same time, right? And these two chromosomes are lined up vertically. Remember the word vertically because I'm going to show you meiosis. It's a bit different. In anaphase, there's a process of pulling. You get one chromatid from your mom and one chromatid from your dad. Telophase is the reverse of prophase, basically. And then you would have actin in the middle right here, splitting in the middle of the cell, forming two identical cells, right? And the results are two cells, two diploid cells. All right, now let's talk about meiosis, germ cells, gametocytes, non-identical gametes, right? They result in four non-identical gametes. The keyword is non-identical. They are all haploids. Excuse me. I'm only gonna talk about meiosis one because that's the most important thing. Meiosis two basically is the same as mitosis. If you look at the book. Drinking water right now. All right, so let's talk about prophase one, right? Prophase one, chromatin. Let me just fix this. All right, so prophase one, same thing, right? Prophase of mitosis and meiosis one, almost the same thing. Chromatin condensed into chromosome, spindles form, but the interesting thing here is the crossing over, right? Tetrad and crossing over. Crossing over is really important for genetic recombination. If you want to learn the detail of the crossing over, you might want to look into the book or genetics. Wu's notes, Dr. Wu's notes might help. Uh, but the book didn't specifically go over, the specific stuff of crossing over. It's related to Mendel's second law. All right, crossing over here, tetrad, right? And then metaphase plate. So that's what I want to talk about here. As you see, in mitosis, I told you that the chromosome from your dad and chromosome from your mom, they are lined up vertically, right? But here they're lined up horizontally. This is interesting because, all right, let me just zoom this. Interesting because the centrioles, the spindles, each spindle is pulling different chromosomes, right? So the left spindle, that'll be right. The right spindle is pulling, let's say this is your dad's chromosome, right? The right spindle is pulling your dad's chromosome, and the left spindle is pulling your mom's chromosome. But in mitosis, each centrioles are pulling this, like your mom and your dad, right? So it's a bit different, right? That's really important because in anaphase, your mom and your dad, you can see the separation clearly, right? They're being separated. In telophase, reverse of prophase, then they get inside cytokinesis happen, and your mom and your dad are being separated, right? They are in two different cells now, and that's a haploid because until then it was a diploid because your mom and your dad were together, but they're being separated, so they're just haploid. The metaphase 2, it's almost the same thing as mitosis. and I'm going to talk about reproduction in the next video. Bye. Yay.